welcome us to. So Sasha, it's great to welcome you back to the Coronavirus Multi-Species Reading Group. It's also a real delight to have your mentor with us today, Vadim, a very senior distinguished virologist. And I think the two papers that we have on the table for discussion today I think are really informative to read side by side. It's kind of two biographical portraits, but also I think trying to get at some of the basic kind of foundational questions of virology. You know, so many of us have encountered viruses because we've become sick, but I think the growing consensus, at least amongst what I would say are the right-minded virologists, it seems like these so-called pathogens are a relative minority of the viral biodiversity that we're exposed to. So it was really interesting for me to read about you know, how these ideas became established. You know, about you know, how viruses change over the course of, you know, infecting a single host, all the dynamic mutations that are occurring, how these ideas about sort of viral recombination got established, once controversial, now established as fact. And you know, we're reading this alongside you know, some of our ideas that we've worked through together, Sasha, about viral symbiosis, and maybe as a counterpoint to some of the things that social theorists like Beth Povinelli have written about the disruptive potential of viral terrorists. So, you know, some of the ideas that I tried to lay out here are, you know, trying to explore the full possibilities of symbiotic relationships. You know, how symbiosis literally means living with, how symbiotic partners can have good, bad, and pluripotent impacts on a host. And maybe, you know, I'll kick it to Sasha. And, you know, we read your earlier piece on living in peace with viruses. Here, I'm trying to, you know, think with that distinguished lecture you gave, you know, about specifically living in peace with coronaviruses. And, you know, after we worked on this together, after, you know, we've talked a lot about living in peace with SARS-CoV-2, do you have any further reflections or thoughts about living in peace?
Yeah, yes, thanks, Eben. It's, yeah, I just also recall when I delivered this lecture and I thought about the title. Idea was mostly I was inspired by my work with poliovirus. At that time, coronaviruses, of course, were not in the limelight like now, although epidemic was there, but it was relatively short-lived, and mostly it was known among scientists, not public at large. But when I was invited to give this talk, so and I thought that how I have to present viruses, I thought that we mostly see them as enemies, so universally, basically. And at the same time, as a researcher, I realized that we know so little about them. And so why this basically approach? Simply just kind of approaching as enemy, although we know that we suffer, but it is universal rejection, basically, without first trying to learn about viruses. And so this is how I came with this title. And it did not change, I would say, over the years. But I was pleasantly surprised that when you pick up on this line, and I learned that you basically independently developed approach to understand multi-species world, starting from completely different background. So indeed, it might be looks like paradoxical, especially now when we know that so many people died and suffered. Yeah, so we're still talking about living in peace. But idea still, we have to learn first. We have to understand that what we deal with, it is probably a subject, not object. So when we deal with as enemy, we kind of, it is faceless. It is basically, we have to eradicate, eliminate. It is enemy. But if you approach and it is subject, it is something you have to understand first. Maybe then we could come up with something more productive and short term and especially long term. So this is probably my current thinking about this.
And again, you know, everyone should feel free to pepper us with questions. Feel free to unmute yourself, write it in the chat, or raise your hand. I might also ask Sasha a follow-up question. So there was an earlier version of this paper where I really dwelled with the idea of the individual. And as I kind of got into the technicalities of what constitutes a viral individual, it ended up being much more complicated than I thought. My initial reading of Sasha's paper, where he describes SARS-CoV-2, you know, I was reading that as an individual, but it's actually, in fact, you know, just one particular specimen that gets abstracted. So I actually might have a really basic question, you know, in a taxonomic sense, or in the sense of like the individual mutant swarm, or, you know, the species and other categories nested within. What is SARS-CoV-2? I mean, a very basic question, but maybe from there you could help people understand the complexities of these different categories of being.
Right. Yeah. So yeah, it does look like a very simple question, but when you try to answer it, it depends again on the perspective. So when we wrote first our paper draft, which was not published, but it's as a preprint, of course, the basic question, when a new infectious disease emerged, and it's known that it's behind this disease is a virus. So people ask whether it's new virus or it is a known virus. And so then you have to distinguish how you distinguish if it's new or old or known virus. And it turned out it is not simple because it depends on your criteria. So what constitutes new virus? So, and what we learned when we explored this in our paper, that most distinguished property which normally we follow, it is disease. It could be modulated with very little changes in the genome of a virus. So sometimes in only one change in genome will change completely drastically how virus interact with the host. And so then question is whether one change would constitute new virus. So for instance, and to put in perspective, because in case of coronaviruses, genome constitutes something like 30,000 letters. So one change in letter would say this is new virus. So it means that we already knew some other virus which was almost identical, but now we have to come up that it is new virus and we have to come up with a new name. So, but if it is not like this, you require more changes. So the question is whether threshold you put when you say again, it will be new virus or it's old virus. And so that is, it turned out that again, it's not easy and we don't have universal system. So maybe people would be surprised, but we don't have a universal system to distinguish new or old virus. And it is made based on the group specific. So in coronaviruses, I was involved in establishing this system of thresholds when we could say, when we assess differences between viruses, we could say if it exceeds threshold, this is new virus. If it's not exceed, so it is not new virus. So in this case of SARS-CoV-2, which was not named, that it's not known whether it's new or not. So we made analysis and we established that this virus, based on the taxonomy, did not exceed its difference with known viruses, did not exceed threshold. It didn't mean that it's old virus. It belongs to a species we already knew. And we named this virus after the species, basically. So that is how we came up with the name. And so name is connected with the novelty in taxonomical sense. So, and maybe to add, it's already kind of talked a long time ago. So when virus replicate in host, it is also important to understand that it is progeny which it generates. It is not identical. It's already not identical. It is already it's mutational swarm. So there are differences in progeny already in one person. And now, for instance, during pandemic, each person who infected contains not a single virus, not a single genomic sequence, although it could be published like single. It is a swarm, and so it is also add complexity to distinguish whether it's how to distinguish between new or old virus.
Yeah, and in part, I might add, this is both an epistemological and an ontological problem. So, the sequencing technologies are basically taking, they're taking little fragments of viruses and, lining up A's and T's and G's and C's. Is that right with RNA? It's also A's and T's and G's and C's, right?
They have three the same, like DNA, but one of them, instead of T, it's U.
Okay, I knew that it was different. All right, A, T.
Yeah, so it's but four
anyway,
letters, yeah. So
base pairs, but it's taking, since there's a bunch of viral individuals that kind of, viral particles, if we're going to really drill down and try to get specific, like a particle each, has its own genome that's probably idiosyncratic and distinct just because of the rate of error with these RNA viruses. So, it gets archived, it gets, after the sequence sort of gets averaged out, they kind of come up with a consensus genome. So the swarm becomes individuated, archived as a digital artifact. But then, trying to think, the paper talks about comparing individuals like Albert Einstein and Marie Curie. I got to think about the individual as a non-local entity. If we're to think about, a bunch of these discrete physical particles as constituting the same swarm of entities that count as an individual, how does that scale up in time and space as individual people contact one another and, jump on transport technologies and end up on the other side of the world? So part of what didn't make it into the paper, because we decided it was too complicated, and I think it needs to be a paper unto itself. I'm going to just read some notes here. So Timothy Morton has described hyperobjects as non-local entities that are massively distributed in time and space relative to the human. While viruses aren't exactly objects, since they have some properties of a purposeful subject, we could think about them as massively distributed biological individuals. So, and where an individual bleeds into these other categories, I think is some, where do you draw the line is, in one way, it's like a question of lumping and splitting, but I would, to use some more of our jargon, Anne-Marie Moll talks about praxeography, so how things are enacted. How do you do sameness and difference in terms of technology? And I think, a lot of the ways that we're doing sameness and difference are either these rapid antigen tests or, tests that are looking at a particular part of the SARS-CoV-2 genome and saying, like, this is identical or this is not, or the whole genome sequences, they're saying this is a strain or not a strain. So yeah, sometimes these differences are consequential in terms of how, they change the movements of us and, epidemiologists and policymakers. But yeah, I think being fuzzy is also important at some levels too.
Right. Yes, so maybe one of the biggest difficulties to contemplate concept of individuum applied to viruses, we don't see viruses unless there is a disease. So it's a disease, it is a manifestation basically of viruses in general perception. Of course, scientists have instruments to visualize, but outside science, of course, everybody else see only disease as a manifestation of viruses. So how you basically apply individuum in this case? And so that is conceptual difficulties. And so in the paper, we define individuums through genetic. And again, it is in view of many, it's abstraction basically, because we're talking about text. And we're saying that, okay, so a single text, so definitive text, genome, it is defined individual. So, and in the same way, we could also define human, although humans, we have a face. So we associate normally human individual with a face. And we cannot do this with virus. But if you think about more broadly, humans also actually evolve, because we don't, every day we are different. And we're different because our cells constantly divide. And so some people say in 7 years, we're completely different. So it's not the same person. But face changes also, but changes slight. But we still apply concept of individuum to this individuum. We know this is the same individual, but it is different individual. And the same with the virus, although we don't see it. And so, and as I said, and you mentioned, we deal with average sequence, because we cannot deal with a definitive sequence, because it would be implied indeed physically to a single virus particle. But normally we deal with a swarm progeny, like 10 power 5 or copies of progeny. And so then we average sequence. But the same with humans. Humans composed with many cells. And cells actually have different genome sequence, slightly different. But we don't think about in these terms. So there are many more similarities between virus and humans if you start thinking about deeply. And so then you could find some parallels which you probably might be surprised.
Yeah, so to do some quick translation there, 10 to the power of 5 is, hundreds of thousands of viral particles is kind of the distillation of this kind of abstract consensus genome, or that gets archived as the individual. And, we don't mutate quite nearly as much as these RNA viruses, but, if there's a Proceedings of National Academy of Science paper about the skin cell of an average middle-aged human, and we might have from 4 to 40,000 mutations that we weren't born with by the time we're middle-aged with these skin cells. Sevgi, you had your hand up.
Thank you. Actually, about this this disease issue, I mean, because we somehow microbes itself also become visible through the diseases, and I was just thinking how to make this invisible positive relationships visible with not just viruses, microbes, because you were mentioning about the research questions that they are starting with positive relationship instead of starting from the ecological relationship. So I was just thinking, what are the implications of them and how we can somehow change these attitudes? Because, for me, for fermentation and somehow microbiome studies was hopeful in that way, but I'm not sure how it's going to be after Corona, because we are still going back this disease approach and making our negative relationship much more prominent than the positive ones, which are probably much more common than disease interactions.
Right. Right. So it is definitely would help if we have more example of the positive interaction, right? So, but I probably already talked about slightly that in my view, it is very difficult to present evidence to study positive interaction. Because for negative interaction, for disease, we have a lot of control. We have kind of normal people or normal host, plants, animals, or something, and then we apply virus and we see changes, negative changes. Something happens. And so this is what people study. But how you study positive? So you need some control and you could see kind of improvement. And so it is a very different setup, because in science, any phenomenon study, you need control. You have to compare with something. And so this is, if you don't have control, you don't have result. Control is critical to any research. And you need positive control, you need negative control. Normally. So this is how framed scientists. This is what I learned from Vadim when I came to the lab. So he said that no control, no result. You could, it is your imagination basically. And so to get evidence for positive interaction, you need completely different experimental setup, which is you have to prove that your control, it is not already positive example. Maybe all of us, which we kind of consider normal, we are under positive influence of viruses, but we simply just don't know.
So Stephanie has mentioned Caitlin Berggren's work in the chat. So we've had Caitlin on twice. So I encourage people to go back and look. She's both done work about, she's an artist who's been living with hepatitis C since she was a small girl. So she's done some artworks about living in peace with this virus and getting past the metaphors of war. But I think this is also a good time to segue into the pursuit of intriguing puzzles. I think this is a lovely article, and I was struck by the note, Sasha, that you initially sent along with the article, which is that there haven't necessarily been a lot of citations for the work that was done with non-pathogenic mice viruses. So, you know, in anthropology and multi-species ethnography, we talk about the art of noticing. And, you know, I think maybe part of noticing is figuring out how to, you know, take knowledges that are you know, not kind of in the crosshairs of, you know, direct human interest, and figure out how, you know, how to elevate certain kinds of findings because they're important to understanding some of the fundamentals of conditions of life. One of the things I was struck with about this paper on page 52 is that interactions between RNA viruses are being described, and at a moment when the molecular tools were much more limited in terms of what could be understood. I really thought it was interesting to learn about not only how a virus is involved in you know, replicating its own genome, but and also protein synthesis, but how one virus can actually do that for another virus. So I would love to hear you or Vadim talk some about those kinds of interactions that we rarely hear about, these sort of ecological dynamics amongst viruses themselves.
Right. Yeah. So Vadim studied in his lab a very broad range of phenomena, including also interaction with viruses. And interaction between viruses, it started actually between progeny of the same parents, because as we already discussed before, one parent give 10,000 or 100,000 progeny. So you have a lot of siblings. Yeah, so in sense, and so they interact already, and so simply this is so it is part of normal replication cycle. We think about very in abstract way, it is look like one progeny and so it replicating. So we don't have in mind that we have so many siblings and they must interact. And they interact on different levels. So interaction could be genetic. So it means that when siblings generated, they so what it is called recombination happens. So that it means that a progeny, so one child could be generated through complex process when it has traits on not a single parent, but two parents. So that is called recombination, because like in like humans, so they are product of two parents, right? And the same applies to viruses. So, but in this case, there is no sex. So there is no male, no there is female. So in this case, there is no difference. But it's still progeny could carry traits of two parents, or maybe even three. So you could call this also interaction. But interaction could be also on the level of products. So for instance, one sibling, one child is something good on one property and another property. And if they were generated separately completely, they would not survive. But these properties complementary. And so each of them helps to each other. When they generated in the same cell, they, as a result, each of them survived because one helped to another. And it is a phenomenon called complementation in genetics. And it is widely studied, and it is classical genetic analysis, and which was established before genomic era, and it would help to understand complexities of interaction. So this is two fundamental type of interactions, even between progeny of the same parents. And of course, when we expand this line of inquiry and include different viruses, or even viruses in bacteria, for instance, interaction also could be something like this, even including recombination and also sort of complementation or some kind of negative interaction. So when one interacting party inhibit another, or even kill, and it's also known. And indeed, what we kind of discuss now, it is happening in the real world, because we tend in the lab study something simplified, and this is understandable to understand cause and consequences. But in the real world, all complexity could happen. And what we witnessed, for instance, in SARS-CoV-2 pandemic, many cases when people infected by more than one virus, type of viruses. It is also kind of normal. This was known before this pandemic.
So I see that Paco has a challenging question in the chat. Paco, do you want to ask it in real time, or do you want me to try to read it and respond?
Well, I can comment briefly, even if you want. I was just interested in hearing your opinion about how Anne-Marie Moll and Timothy Morton's work that you have just brought in the discussion have what the concepts of these authors have to do with this contrast between the processual philosophy of biology views that have been already discussed in the reading group, when John Dupré, for instance, has presented, or Stefan Gutting, with the more traditional taxonomic views, no, that sometimes are, as Alexander was saying, very important to continue to practice in the context of modern contemporary molecularized and bioinformatic virology and other ways of doing microbiology.
Yeah, so I think I'm going to lean on an earlier paper that I wrote about species, where I was explicitly using the work of Anne-Marie Moll to think about how species are enacted. So in that paper, I was thinking about very different kinds of life. I was thinking about frogs and plants and fig trees, wasps that pollinate fig trees, and then Wolbachia bacteria that sort of influence the breeding of these wasps, basically looking at complicated multi-species entanglements that were being mapped out by taxonomists that looked at practices of recognition, differentiation, and categorization, not as they're being imposed by people as we're trying to puzzle through these different categories, but trying to figure out how organisms recognize one another. I think we're in part of that paper was also very much engaging with John Dupre, who you just invoked and who's been a visitor here, to think about what he calls promiscuous realism, to insist that one sense of a species doesn't work for all organisms. The idea of a species that you develop for a plant or a mammal really breaks down if you try to enforce that on a bacteria or a virus. And I think doing that work, or thinking with promiscuous realism, involves looking at the practices of people like Sasha who are deep in the weeds of trying to figure out what these different forms of life or living processes, to again invoke Dupre, are all about. I would point to in trying to, I'm not sure I'll be able to bring Morton in, but I might try. But I would point to ways, again, thinking praxiographically, that the consensus genomes are established, the ways that these different kinds of viruses get archived, but then also the intellectual work that people like Sasha and colleagues did to determine, as he was talking about in the beginning, how a form gets assessed. Is this a novel form, or is this something we've seen before? And then mapping out these very complicated relations, which in the realm of viruses, more so than say plants or mammals, a lot of this is dependent on algorithms and in alignments of genomes and computational methods. So I think one of the interesting things that Ian Hacking identified a long time ago is the ways that categories loop in the world. That if you recognize an emergent category, that can influence reality as that category comes to inform, say, new technologies of differentiation and recognition. So after Sasha produced this influential paper, now we have a proliferation of tests that in one way or another claim to know whether SARS-CoV-2 is present or absent. And those tests then have the looping effects that Hacking describes. Getting a positive or negative result changes how we move through the world. Even those tests, the antigen tests, for example, are only going to detect certain levels of virus. They're not going to give you a quantitative result like a PCR, some of the PCR tests do. But many people argue that having a negative antigen test is enough to influence how you move through the world in an epidemiologically or clinically meaningful way. I'm not sure I brought Morton in yet, but I think I tied in some. So, and do you have a follow-up on Morton?
No, well, actually, this was really an intriguing way of bringing together hyperobjects into this discussion. I had never thought about it, so I was really curious about what to do with that. No.
Yeah, I mean, maybe if I want to just kind of think on the fly here, maybe all species are sort of hyperobjects. So if we're talking about, say, an insect, you put a voucher specimen in the museum that's covered in gold. That's called the holotype, and it's kind of it's a synecdoche. It's a part whole kind of representational modality. And I think maybe these individuals that get the consensus genomes that get kind of, distilled from the cloud of a mutant swarm, maybe that too is kind of grasping at this hyper object that has, ultimately pretty fuzzy boundaries, because if you look at species in time and space, they kind of diverge and converge, and yeah, this might be totally abstract.
But then when you have introduced hacking,
yeah,
maybe Morton's idea looks a bit limited, no? Because it doesn't really include this constructivist aspect, no? Or what do you think?
Yeah, indeed. I mean, I don't claim to know Morton's work that well, so maybe there's other people on here who've read the whole book. I've just kind of dipped into it. But yeah, I get a sense that hyperobjects are maybe a little less, yeah, I mean, they're objects, they're not subjects. And maybe this is where I'm trying to kind of make a move in response and say that the virus isn't an object, that it's, it's a subject and, a multitude of subjects. And, it's ultimately intersubjective, both in terms of, how the virus interfaces with the host, but also how these populations of viruses interface with people like Sasha who are trying to understand, the dynamics of the populations.
Exactly.
I saw Sasha nodding.
That would make it more coherent with Sasha's position as presented earlier.
Sasha, I hope that wasn't too much jargon, but I'd love to hear your thoughts too.
Right, yeah, it's fascinating what you're talking about. I'm sorry. Also, I did not read this book and original. So it's and they're probably far away from what my expertise, and so that I'm not completely sure that what I could comment would be to the point. But indeed, for me, it is important to distinguish what we're talking about subject and object. It is critical, right? So divide. So because if you're going to subject, so then, for instance, when we're talking about species or something, you would deal completely different ways than with object, right? And so then important step, if you name it. So if you name your, it's your relationship, it is completely changes. So you recognize that it's not as one of the kind, but it's individual. And so that is, this is a very important step. And this is why, for instance, if we recognize as viruses as objects and we name them, and then we respect this name and interact with with another kind, so you bring this a completely different field of interaction. So you have to deal with completely different than with, for instance, table or chair or something that you could dispose, and so something that is absolutely different type of relationship. If you kind of deeply look in viruses and in human beings for comparison, because it's much easier, because everybody knows what is human, you could see many more parallels that you could imagine. So of course there are differences. So humans, it's like physical object, right? So it has boundaries and everything, but in a way it is ecosystem. We know that it's consistent so many different cells and also different organisms. We simply tend to associate it with a face. And this is how kind of normally we deal. But in reality, it is ecosystem. It's a huge ecosystem which is constantly evolved, but it is the same with viruses. It's maybe not physically attached to each other, and so they distribute it in a way. But in many other connections, so there are parallels. And also, when we're talking about mutation rate, it's also important point that generation life in viruses is much shorter than in humans. And that is critical. So mutation rate, if you normalize it, on genome, it's the same in humans and in viruses. So difference is that progeny is generated every 10 hours. And in humans, it is 30 years. So over 30 years, you have, I don't know, I don't kind of like 40,000 generations in viruses. But it is completely different scale. And of course, then you have a mutation. So mutations are generations. But also in humans, we have generation in 30 years. But if you look on the cell level, like you mentioned, it is much more frequently. And so then what is generation? Of course, children are given like every 30 years. But on the cell level, it is much more frequent. And so then difference between how you call this distributed or something processual, so it could be applied to humans.
Thanks, Sasha. Rachel, you've got your hand up.
Thank you for this lovely paper and lovely conversation. I was just thinking about that segment that begins on page 8, where you're introducing Charlotte's or citing Charlotte's work on symbiosis and as living together and symbiotic partners. And just it's such a lovely kind of walk down memory lane too of so many of these beautiful thinkers and writers that have joined us to date in these sessions with us. But I was wondering if I could hear you riff a little bit more, Eben, on that careful tightrope that you're walking in this piece about kind of. I think there's such a subtle and careful attention to the collectivity and possibility and plurality of living together. And at the same time, you're walking such a careful tightrope in this piece about the kind of the political, I guess I could call it a political potency or a political it is a kind of terrorism, I think, of framing the living with as a way of sort of leaving majority and minority experiences, dividing them, and then sort of leaving some people behind, I think, is what's happened, what we're seeing, I think, in a lot of political and economic framings of these. And there's such an important difference that you are framing in this piece. And so I would just love to hear you riff a little bit more on that last segment of the paper as it's currently written, where you're kind of distinguishing all these beautiful thinkers and enveloping this concept of collectivity and living together and plurality. And you're using terms, you know, swarm and melting and clouds and these kinds of concepts with the tension that you're also framing about this sort of the way that living with gets framed as just leaving, well, you know, the majority, you know, experiences X, therefore, you know, we can sort of leave others behind. So I'd just love to hear you riff and say more.
Yeah, so maybe I'll just start, you know, kind of with a little history of ideas here. So as I introduce symbiosis on my version of the paper, it's bottom of page 9, through Charlotte Brevis, you know, living together in situations that can have good, bad, or pluripotent effects on hosts, but also in the context of this idea of symbiogenesis that was first articulated by the Russian botanist Konstantin and Sasha, apologies, I'm going to butcher this last name, but Mira Tchaikovsky in 1910, and that was to think about the origin of photosynthesis as plants as acquiring free living microorganisms, cyanobacteria. So Donna Haraway mobilizes this idea of symbiogenesis in the 1990s to think about the experience of people with AIDS. And Donna was, you know, you could read that as a hasty celebration of life with a virus, but it's also written from the standpoint of someone whose husband has just died from AIDS. And so Haraway was trying to think about symbiosis in not only a biological sense, but in a sense of the uneasy alliances that people who were living with a virus that at that time, there wasn't a medical intervention, but even to enter that promissory space of a possible set of research futures that would bring medical interventions within reach. This involved these alliances with scientists, drug companies, lawyers, mayors, the mayor of New York City famously, journalists. And I think we can dig down into that moment in the HIV advocacy world to think through some of these more complicated politics of living with that involve a minority and a majority. So I think the early HIV activism community had really two different groups. There were some that were very much focused on the science and doing everything that they could to get better, more innovative, risky science, to put AZT behind them and basically demand that they become experimental subjects in ever more risky, venturesome experiments. And then there was another group of the HIV advocacy community, the AIDS advocacy community, that was more concerned with who was getting AIDS, how and why, thinking about how insecure housing, how racism, how homophobia were all informing the uneven life chances that people were experiencing back then. And now fast forward 20, 30 years, now that there are good medical interventions, in some ways like the earlier hopes that some kind of dismissed as kind of pie in the sky, you'll never get a cure for HIV, those have come into being. So on the one hand, it's the success of that one group, but the activists are still trying to, within the realm of HIV and AIDS, trying to advocate for members of the community that are still experiencing insecure housing, that are still having difficulty living with ongoing discrimination because of their viral status, or because of the complicated ways that race and class intersect to produce unequal health outcomes. So I profiled one of these people in my latest book, Jay Johnson in Philadelphia, who leads the volunteer program in Action Wellness. And his organization in a country where you're not guaranteed healthcare, as you are in a place like Australia or many parts of Europe, he's able to, even in that landscape, guarantee that people with long-term conditions like HIV are able to access medicine. But then, so Ben, who's on the Zoom call here, Ben Hagarty, and many others are starting to really look at the uneven life chances on a global scale, how these global inequalities are playing out with HIV. And there's so many complicated ways that privilege is playing out in this pandemic. I mean, we can all point to, and we've kind of done so many different dimensions of this with this reading group. We've heard Carrie Fresher's work, Tony Hatch and others talking about this emergent category of essential worker that was basically expendable worker, but also essential for the rest of us who had the privilege of sheltering in place. We've dealt with things related to invisibility and visibility, as the pandemic became hyper visible in places like Italy and the US, even Brazil, other parts of Latin America, other parts of Southeast Asia, this mass death was invisible. And there's been a very uneven rollout and very complicated politics of, who even wants a vaccine in places that have, experienced military genocides. And I saw that there was a question in the chat. Let's see if I can pull it up. Lots of questions in the chat. I'll stop talking because I've kind of I think I've touched on some of the things that Rachel mentioned, but I see a question both from Melanie and Paco. So Melanie, do you want to jump in?
I mean, it's more a comment. And I find that in a lot of papers I read these days, when it comes to this discussion of subjects, objects, treating the non-human world as objects, or well, making a point for subject status, there's this like flagging, like, yes, we're aware that in many indigenous knowledge systems, the individual doesn't exist as it does in the, it's not like framed in the same way as in the dominant Western scientific knowledge system. Like every being is connected, and it's like a hyper subject connected across species, but also connected across time. So, I'm just wondering how that can be brought into the discussion. But maybe we just need more people studying like viruses specifically. I don't know if you are aware of people doing that already, or
yeah, super interesting.
I mean, it's because it's connected to, I think it's very much connected to the what you write about in the later part of the chapter, the politics, the privilege that you spoke about now, which was like it's kind of maybe far-fetched, but connected to the politics of knowledge and how this more like pluriverse epistemologies have been marginalized, but kind of now are like popping up again, because they've always been pluriversal. While we just in the West, we're just catching up with what people in the South have known all along and never forgot.
Super interesting. Yeah, I like that. I think you just maybe coined a new word, hyper subject. Paco, I would respond, but we only have a few minutes. And as I was hearing you talk, I was thinking we should, be reading Marilyn Strathern's work on relations alongside texts about viruses. But so many other Indigenous theorists too. I mean, Kim Tallbear has a lovely piece in a book that we have coming out later this year. The book is about multi-species justice, and in some ways she's kind of pushing against the frame of multi-species ontologies to think about relations and living with relations that are sometimes good relations and sometimes bad relations. Like in communities, you have all kinds of complicated, fraught relations. And so yeah, let's have, I think Kim would be a great one to invite into the group as well. And Paco, do you want to, you've got a comment here about symbiogenesis?
Just very briefly, I think it's good to remember, as you have invited us to do, even that symbiogenetic perspectives are essential, not to the argument that you developed in your paper. And I was just recalling niche construction theory and the ideas of Eva Jablonka and Miriam Lamb about evolution in 4 dimensions to perhaps just put it together, with the symbiogenetic perspective. I think that Donna Haraway sometimes actually does that. And actually, once you have done that, to include the viral world as part of that, which usually is forgotten. When people, for instance, talk about cultural evolution, in the context of these biological theories, they never really talk about viruses, and I think that this is necessary now to do. And Sasha's work, I think, has made me think seriously about it. So thank you very much.
Thank you. And some of those names are new to me, so I'd love to stay in touch and get some of those references. And so this has been an amazingly generative discussion for me. And I should mention, after I wrote this paper and one other one, I just realized I could start writing a book. So I've started writing a book as of last week, provisionally called Life in a Pandemic. I'm trying to rethink ideas of life, using Agamben as a sort of I'm not going to punch on Agamben like everybody else has done, but kind of unraveling some of his foundational ideas about life. But thank you. And Petronella, do you want to jump in with your comment here too? You're saying about relational context.
Yeah, I was just going to say that, of course, thinking from the point of view of Indigenous people in Central Australia, individuality or a focus on that, but the positionality that within a relational context is different. And I think that's a really interesting thing to think with in terms of going back to the individual and the swarm.
Yeah, indeed. Sasha, do you have any final thoughts?
I just thank you. Yeah, I also enjoyed it very much, and thanks for the time I received to talk about. So, but then I just a small comment, short comment about relationship. So during pandemic, we introduced in many parts of the world lockdowns, and they called social distancing, right? So, but in fact, it was physical distancing in scientific literature, but it was converted to social distancing. And why? Because in reality, it affected our relationship. Of course, it was physical distancing, but it was, and like what you mentioned, relationship could be bad, good, and nothing, but it is relationship. And we are kind of society, kind of human being, we dependent on relationship. And this lockdown, it of course was for the purpose, for specific, to limit basically spread of the virus, but it affected all relationships. And so that is, of course, we still have to count impact of this intervention.
Thanks, Sasha, and thanks for being here. And Vadim, it was a real pleasure and an honor to be in your presence. It's an amazing opportunity to have this intergenerational fount of knowledge here. Thank you all. It's great to have so many of you in conversation. This is, one of the longest conversations I've ever had. It's lasted more, I guess, almost 2 years now. So, thank you all for coming back. And Rachel, who's on the deck for next week? I can't remember.
I think we've got Anand, but it isn't for a couple of. It's March 7th. Is that next?
March 7th. So if y'all haven't noticed, we're going to not every week, but every other week. So Anand Pandian is coming up next. All right. Thanks so much for coming, and be in touch.
Yeah,
Thanks,
you.
everybody.
Thank you. See you next time.
Thanks.